Hello, welcome to this channel again. Today we're going to discuss about the endogenous antigen presentation by MHC-I molecule. Yesterday, the last video was about the exogenous antigen presentation by MHC-II molecule. Now, for a basic idea of this video, endogenous antigen presentation means presentation of an antigen which somehow managed to enter in our cell. Okay, so this antigen presentation is solely done by MHC-I molecule. The MHC-I molecule is present in all nucleated cells, including the antigen presenting cell. Okay, so this pathway is also known as cytosolic pathway of antigen presentation. Okay, here MHC class 1 molecule is solely involved and which is present in all nucleated cells. So it involves several steps, which I have mentioned in very easy way. Okay, so an antigen enter in our cell. The first stage is antigen entering in our cell. The second stage is processing of antigen. The third stage is antigenic peptide formation. Okay, after processing, the antigen are degraded and antigenic peptides are formed. And the fourth stage is presentation of antigen by MHC-I molecule. So how this processing takes place, how the cell knows that this antigen need to be degraded and antigenic peptide need to be formed. So there is a unique protein known as ubiquitin protein which tag the antigen which need to be degraded, and there is a protease complex known as proteasome which degrade the tagged antigen with ubiquitin protein. This protease complex known as proteasome uses ATP to degrade the antigenic peptides. There is a uh, there is a protein known as TAP transporter associated with antigen processing, which transport the processed antigen to RER or the rough endoplasmic reticulum. Here mentioned that TAP protein is made up of two components. First is TAP1 and TAP2. So TAP protein is a complex protein made up of two subunits, TAP1 and TAP2. Now let's see the process of how the antigen presentation is taken place by is carried out by MHC-I molecule. Okay, we have already discussed that the TAP protein transport the antigenic peptide to endoplasmic reticulum. Okay, here it is mentioned that peptide enter the endoplasmic reticulum and bound with MHC-I molecule. Okay, so there is a chaperone, molecular chaperone. Chaperone means a protein that fold the newly made protein in a way that it can be active. Okay, so there is a chaperone known as calnexin which bind to the alpha chain and fold it to its active form. Okay, alpha chain of MHC-I molecule is made in endoplasmic reticulum. A chaperone known as calnexin bind to the alpha chain and fold it to its functional form. Okay, and after that beta chain bind to the alpha chain and calnexin is released. So second, now calreticulin and tapesin, which are two protein, bind to alpha and beta chain and bring the antigen associated with tap near them. What the After releasing of calnexin, two protein, calreticulin and tapesin, bind to the alpha chain. Okay, bind to the alpha chain of MHC-I molecule, and they attract the tap protein, which bring the antigenic peptide in the endoplasmic reticulum, is attracted by the calreticulin and tapesin protein, and they bind to the MHC-I binding cleft. Okay, now the antigenic peptide bound to the MHC-I binding cleft, and MHC-I release from calreticulin and tapesin protein and move out from ER and enter the Golgi body. Okay, then the MHC-I bound with the antigenic peptide exposed in the membrane of the cell and present the antigen to the CD8 plus receptors of cytotoxic T cell. Okay, let's understand this with a diagram. See, it is endogenic. It is the endogenic See, it is the endogenic antigen. Okay, endogenous antigen. Now, these secret some proteins. Okay, now there is no any mention of ubiquitin protein and other complexes and the calreticulin and calnexin. So try to understand. Now here, okay, here the pathogen enters. This pathogen is tagged with this pathogen is tagged with some ubiquitin protein. Okay, ubiquitin protein. Now this tagged antigen enter to the proteasome complex. Okay, proteasome complex, and this proteasome complex degrade degrade this pathogen and produce antigenic peptides. These antigenic peptides are uh, carried out by TAP protein and enter the endoplasmic reticulum. In endoplasmic reticulum, there are various proteins like calreticulin and tapicin, which uh, which attract them towards the alpha and beta chain of MSC1 molecule. And after this, the antigenic peptide bind to the binding cleft of MSC1 molecule. Now the MSC1 molecule is ready to present the antigen. Okay, this MSC1 molecule goes from endoplasmic reticulum to the cell membrane of the cell of the nucleated cell and present the antigen in membrane to the CD8 plus T cells or cytotoxic T cell. It is the receptor of cytotoxic T cells, and it is the MSC1 molecule, and it is the antigenic peptides. Okay, so we already know that the MSC1 molecule always present the antigen to the cytotoxic T cells, and the MSC2 molecule always present the antigen to T helper cells. Thank you.